I'd like to begin by welcoming you on behalf of Carla, Nestor, and myself to the University of Iowa. For some of you, this is your second, third, fourth, fifth visit. For some of you, this is your first time. The MORL, and there's a huge MORL team, and if they're in the audience, could you please stand? I'd like to recognize them for all that they do to make this possible for all of you. The MORL has 40 research scientists, and we work for you. And we work to understand this disease better. We work to change the landscape in the treatment of this disease. We work to change the knowledge base of this disease so that all of you can have better outcomes and better care. Now, when I was a high school student, decades ago, they used to have the Cliff Notes. So are all of you familiar with Cliff Notes? Kind of the cheater's guide to everything, so that you could kind of get the nitty-gritty stuff without doing all the work. And so what I want to do today is to lay a foundation for you about complement and the Cliff Notes guide to complement to make things easier for you to understand. Complement is foundational to your disease, and it's complicated. And so if you drift off during the next 20 minutes, that's fine, but remember this slide before you drift off. So this is a slide that Carla Nestor made, and she calls it her peacock. And what you can see is we have atypical aHUS here in the middle, and at the bottom we have complement-mediated aHUS or complement-mediated TMA. So many of you, statistically about half of you diagnosed with atypical aHUS in this room, have a complement-driving variant that Nico from North Dakota will explain to you about next, have a complement-driving variant in a gene that's causing your phenotype. But as we've learned more about atypical aHUS, you can see that there's lots of other drivers of disease. There's lots of other triggers. And how these triggers relate to complement or how these triggers relate to coagulation is currently something that we're actively studying. So remember this, that it's become more and more complicated. And also remember that there is crosstalk between the complement cascade that I'm going to review with you and also the coagulation cascade that probably most of you are not that familiar with. So what is complement? So I'm going to talk a little bit about the complement system, why things are named, why it's called a complement cascade, what provides the control, what are biomarkers. So some of you gave blood today, and without asking you, we can study your blood, we can run some biomarkers, and I can tell you which ones of you are on Solaris. And so that's called biomarker profiling. So we know if you're on Solaris, we know if you have active disease, and we don't need to ask you. And that's because we can look at these complement pieces like pieces in a puzzle. We can turn them over, we can fit them together, they interlock, and they help us make an individualized, personalized picture for each of you. And then I'll talk briefly about atypical aHUS, why, if you have this diagnosis, you attack your own cells, and then I'll conclude with complement and coagulation and crosstalk, and really what is that. So the complement system is like a head coach. The complement system is the cornerstone of what we call innate immunity. So basically, all of us have two types of immunity. We have innate immunity, what we're born with, and we have acquired immunity. When you go get your flu shot, how many of you had your flu shot? If you had your flu shot, that's acquired immunity. They expose you to something, and you develop antibodies to it. But the moment you get a cold, the moment you get an infection, your complement cascade revs up. It's the sentinel that's circulating in the blood all the time. There's huge amounts of C3, there's huge amounts of factor B, and there is so much that complement can be triggered immediately and yet under exquisite control. And so it is the cornerstone of innate immunity. It's like a carburetor, it's a disposal system, and it talks to adaptive immunity. So when you get your flu shot, it talks to the B cells that are making the antibodies to help you when you get your flu shot. And so in innate immunity, it marks cells for destruction. It helps cells and makes them explode. It calls in helper cells, and it also calls in molecules that lead to inflammation. So Solaris, not only does it stop the complement cascade, and we'll see where in a moment, but it also prevents generation of C5a, which you've all heard of, which is one of the most potent inflammatory molecules in the body. So you're getting a double whammy. You're getting rid of inflammation when you're on Solaris, and that's why for some of you it may help if you have, for example, Crohn's disease. And you are getting rid of the terminal complement cascade. You're stopping the complement system. It's a disposal. Anybody know what movie that's from? Yes. Anyway, so it marks cells for destruction. And then it enhances the antibody response. So when you get your flu shot, it enhances that response so you get a better outcome. It talks to T cells and it enhances your immunological memory. So when you've been exposed to something and you've had a cold, if you get exposed to the same thing, CMV for example, if you get exposed to the same strain, and your body will remember that, and so then the sickness that you have, if any, will be markedly reduced. So how are things named in the complement system? It's totally confusing. There are 3 initiating pathways. So think of it as a tree. And the branches are the initiating pathways, and the trunk is the terminal complement pathway. And so there are 3 initiating pathways, the classical, the lectin, and the alternative. And they feed into the trunk, which is the terminal complement pathway, and that's, again, where Solaris stops. So Solaris stops everything from propagating down. So the classical pathway was actually described first, and when scientists discovered the proteins that make up the classical pathway, they named them with the letter C for complement, and then a number. And that number was in the order not of activation, but in the order of discovery. And so that's why things are not sequential when you talk about complement activity. So it goes C1, C4, C2, C3, and then they got it right, 5, 6, 7, 8, 9. But that mix-up was because things were labeled in the order that they were discovered. The alternative pathway was discovered after the classical pathway, and at that time they changed the classification system and they used letters instead of numbers, and it was called alternative again because they thought it was an alternative way to activate complement. And this is pertinent to all of you in this room. The alternative pathway is what we call constitutively active. So in each of us, all the time, the alternative pathway is on. And it's on at a very low rate. It's almost like a dripping faucet. So in our bathroom, one of our faucets leaks and it drips slowly. And that's what the alternative pathway does. And that's actually called tickover, so it has a name. And about 2% of C3 ticks over in the alternative pathway all the time. Now for some of us, the tick over is a little bit faster. Some faucets drip faster than others. And in those persons in whom the tick over is faster, the likelihood of getting some complement-mediated renal diseases is increased. And then the most recent pathway discovered is the lectin pathway. and its name nomenclature is even different. And the lectin pathway, like the classical pathway, feeds into C4 and C2. Both the classical pathway and the lectin pathway patrol and look for specific protein signatures of foreign invaders. And so the classical pathway looks for antibodies attached to cells, and if it sees that, then complement is revved up. And the lectin pathway looks at the sugar signature on bacterial walls and recognizes that signature as different from your own cell signature, and so those bacterial cells are attacked by complement, all the while preventing any damage to your own cells. So you'll notice a couple of things. First of all, the activation is sequential, and because it's sequential, it's called a cascade. There are 3 initiating pathways, and there's an amplification portion. So there's an initiating portion and an amplification portion. The amplification portion generates more of what we call C3 convertase. C3 convertase is the most important enzyme in the complement system. It is so robust that over the course of a few minutes, 10 billion molecules of C3 can be consumed, generating massive amounts of C3 convertase. So that gives you a flavor for how powerful the complement system is. And if you have any doubt about it, you were diagnosed with a disease that was caused by dysregulation or abnormality of the complement system. And that's a very vivid example of how powerful it is and what can go wrong when something does go wrong. So, if you have a complement system, you need brakes. And there are brakes to the complement system. And the brakes, the most important brakes are factor H, which many of you are familiar with, and factor I. So I'm showing you an example of 2 brakes. Factor I does not work alone. Factor I works in concert with factor H. And factor H provides almost a landing dock for factor I to dock into so that it can help with the degradation of C3b. And so if the brakes work properly, and if complement is working properly, again, we have massive amounts of C3 in our circulation, massive amounts of factor B in our circulation, all ready to mount a complement attack on a bacteria if you get infected. All the while, and as this attack is mounted, in a sense, it's a little bit indiscriminate. So it's attacking a bacteria, but a bacteria might be right next to one of your own cells. And so all the while, to control this, your own cells are able to attract factor H. Your own cells have cell-bound regulators of complement. And so the complement attack is directed to the bacteria, but not to your own cells. So it's totally cool the way all that works. So what is biomarker profiling, and how can we figure out if you are on Solaris, or if your disease is smoldering and a little bit active, or if your disease is rampant, or if your disease is quiescent? So we can decide that by taking all of these. So each of these we can measure in the lab. We can measure each of those protein constituents of the complement cascade. Each of them have what we call cleavage or breakdown products or split products. Those names are interchangeable. We can measure the little bits that are present in your circulatory system. We can measure the enzymes, C3 convertase, and its functional consequence. We can measure C5 convertase and its functional consequence. Think of all of those as pieces of a puzzle. And think of a puzzle where you have lost the top of the box and you have no clue if it's a swan, if it's a cornfield. You have no clue what the picture is. And you do what everybody does when they put a puzzle together. You turn over each piece, you put all the same colors in one pile, you put all the edges in another pile, and then you start putting them together. And that's exactly what we do. Some of the pieces, some of the information that we get is uninformative and it doesn't help us at all. But we look at everything. Some of it interlocks. And the pieces, just like pieces of a puzzle, the results talk to each other, they confirm each other. And that's what we do in all of you, and that's how we're able to figure out what's going on. And sometimes we have an incomplete picture. Sometimes we just have an edge. Sometimes we've got 25% of the puzzle done, and we call you up and say, well, Mr. Bierman, we have no clue what's going on. We need to turn over some more pieces. We need to do some more work. And then we do some more work, and 2 years later, we say, well, we figured it out. We've got a good idea about what's going on. For some of you, we can do that in exquisite detail. For others of you, we can't. And it doesn't mean that we don't know what we're doing. It means that we haven't turned over enough pieces of the puzzle. We haven't asked enough questions. We haven't gone down enough roads to get a better idea of what's happening at an individual level. And that's what we try and do to help each one of you. So now you kind of understand the background. And so I'm going to focus on a little bit about what happens in atypical aHUS. And in atypical aHUS, in the normal system, as we talked about, complement is circulating, the proteins are circulating. They recognize the bacterial cell surface and they rev up on the bacterial cell surface. And so they make the C3 convertase, which is C3BBB. They make the C5 convertase, and they explode the bacteria. And that's a huge, that's phylogenetically. So that system of defense has been with us for ages. Many, many, many animals have this kind of system of defense. So now, when your complement system is healthy, remember I told you could have a bacteria right next to one of your own cells. The reaction is indiscriminate, but the brakes recognize your cell surface and they say, whoa, we don't want the C3 convertase working on this cell surface because that's part of me. And in atypical aHUS, that recognition is impaired. And that recognition is impaired at some level if you have complement-mediated atypical aHUS. And the impairment can be in one gene, it can be in factor H, the impairment can be in factor I, the impairment can be in C3, the impairment can be in factor B. And while all of it has a common final outcome, which you see here, knowing where the impairment is can be beneficial to you in knowing how to manage your own personal disease. So here's an example of factor H. So factor H, as Linda knows, is a necklace, a pearl necklace with 4 white pearls at the end and 16 black pearls. And these pearls, the end ones are like magnets, and they are magnets that recognize your own cells, they stick to your cells, and by sticking to your cells, they can control complement activation on your cells, and they can stop it. So if that magnet is broken, if that stickiness is lost, then you lose complement control on your own cells. That same thing can happen if you have variants in MCP. So MCP is another brake to halt the complement system. It's a different brake than factor H, but it's a brake nonetheless. And so what we ask ourselves is, in each of you, is there something wrong in one of your brakes? And which brake is it? And how does that impact your personal disease?
How the MCP brake works different from like the factors?
Yeah. So the MCP brake is so some brakes are circulating. So some brakes are in the circulatory system and they're floating around with C3 and factor B. MCP is one of the cell membrane bound brakes, and so it is stuck on the cell. It is not in the circulatory system. It is not made by the liver. So factor H, most of the proteins that I've been talking to you about are made by the liver. Liver makes them, squirts them out, and they circulate throughout your body. MCP is on cells in the kidney. It's on cells in your circulatory system. It's on cells in your liver. So it's on a bunch of cells serving as the same purpose as factor H, but it's a cell-bound sentinel that is acting to control complement in that microenvironment. And there's a number of other cell-bound regulators. And so what happens is if your brakes are not working, or if your complement cascade is revved up so much that it overcomes the brakes, if you're driving your car too fast and you're too close to that guy ahead of you and he slams on his brakes, your brakes, even if they work, are not working good enough because you're going too fast and you're going to hit him. If your complement system is revved up too much, for example, by mutations in C3, then this system on your own cell surface loses control. And if it loses control and its cell surface lost control, you have atypical aHUS. You have complement-mediated aHUS. If you have loss of control in the circulatory system, you have its kissing cousin, C3 glomerulopathy. So they are two complement-mediated renal diseases that have a different pathophysiology in the level of dysfunction, but share a commonality in the dysregulation of the complement system. So how else can you ruin a brake? Well, so if your brakes, if your brakes are shielded, and so this is an antibody, then factor H will not work. And so if you develop an antibody to factor H, and an antibody is a protein that sticks on another protein, by sticking on factor H, it may cover up the essential portions of factor H that are necessary for it to work. If it's covered up, it can't work. It's like you don't have any brakes, and you end up, again, with atypical aHUS. So some of you in this room have antibody-driven atypical aHUS. This was called by Peter Zipfel DEAP aHUS, D-E-A-P aHUS. We know it's associated with the absence of a gene called complement factor H related 1. In this room, there are about 3 or 4 people who have 0 copies of factor H related 1. There's about 20 in this room, 20 to 25, who have only 1 copy. That's because it's a frequent variation amongst people. If you happen to have 0 copies, you are at risk for developing these antibodies. If you develop these antibodies, then you can develop atypical aHUS. So that's important, and that will become increasingly important as the years go by, because it is knowledge like this that directs therapy, that directs the length of therapy, that directs alternatives to therapy. The more you know, the better informed you are, the better you'll be able to advocate for yourself or your loved one. There's also DGKE, and I think Boo's going to talk briefly about that, which is not a complement gene, is associated with atypical aHUS, and note in the box here that lack of DGKE causes too much clotting. So now I'm not even talking about complement, I'm talking about clotting, and if I'm talking about clotting, I'm talking about coagulation. And so I'm going to introduce to you the concept of crosstalk between the complement system and the coagulation system. And that means that these systems don't exist in isolation one from the other, but rather they're intimately connected. And so here is the complement system showing you the alternative pathway, the C3 convertase, C5 convertase, and that's what we've been talking about for the past 15 minutes. Here's the coagulation system, which is as complicated and which many of you probably haven't thought much about at all. But the two systems talk to each other. So these proteins in the coagulation system can also cleave, they can also cut C3. If they cut C3, they can affect the activation of complement and they can affect your disease profile. So we have good evidence that if you have an increase in coagulability, if you're susceptible to clots, you're more likely to ramp up your complement system. If you ramp up your complement system and you're predisposed to atypical aHUS, you're more likely to get atypical aHUS. So there's an exquisite relationship between these, both in the pathophysiology and, as Boo will touch on, what we think can be alternatives or possibilities for therapy. And similarly, these four proteins also cleave C5, and if you cleave C5, you get your C5a, which is a very powerful inflammatory molecule, and you start the terminal complement system. And this shows the effect of these in cleaving both C3 and C5. So I hope you've taken lots of notes, and maybe some of you have done some doodles. So thanks for your attention.